Hello everyone, and welcome to Biopedia. Barring exceptions such as trisomies, humans have 46 chromosomes split into 23 pairs, 22 pairs of autosomes, and 1 pair of sex chromosomes, either XX or XY. This might not be something you've ever thought about. However, different organisms have different chromosome numbers. For example, while humans have 62 and 64 in total, the chromosome count for donkeys or horses is 62 and 64 respectively. Looking a bit closer to home, great apes, referring to gorillas, chimpanzees, orangutans and bonobos, have 24 pairs of chromosomes, making for 48 in total. So why this difference between humans and our phylogenetically close cousins? The short version is that the human chromosome 2 is actually the result of a fusion between two previous chromosomes, chromosome 2A and chromosome 2B. This fusion of chromosomes 2A and 2B took place a few million years before the present, and it can be localized to a section of chromosome 2 known as 2q1. So, how can we tell? Well, a region of this chromosome called chromosome band Q13 is known to contain some DNA from telomeres, despite being within the chromosome. Now, this is not a summary about telomeres, that's probably for a future episode, but the important point for our purposes today is that telomeres, consisting of long repeats of the sequence TTAGGG, are located at the end of chromosomes, kind of acting as a protective cap. These repeats can also be found at what are known as subtelomeric regions, which are located next to telomeres. Notably, telomeric DNA can be detected in this specific 2q13 band. Given that telomeres are usually located at the tips of the chromosome, this points to an event in evolutionary history where telomeric DNA somehow ended up inside the chromosome. A 1991 study, using a library consisting of human genome cosmids, managed to identify 2 cosmids within 2q13 which each contained 2 telomeric repeats, arranged in such a way that the tips of the telomeric DNA were facing each other. I'm going to gloss over this a bit, but in brief, a cosmid is a hybrid form of plasmid which stores an average of 45,000 letters of DNA, which can subsequently be cloned into a new organism. For the study in question, lots of cosmids with different stretches of DNA, which made up a cosmid library, were used to identify the relevant ones. How this study achieved this is that the cosmids were selected which specifically contained both repeats of TTAGGG and PSC4. Why is this relevant? TTAGGG repeats are what telomeres essentially consist of, as discussed above. In addition, PSC4 is a section of 300 bases which was known to localize at subtelomeric stretches on the chromosome, and also at this 2q13 locus. Cosmids which contained both of these stretches of DNA were hybridized to chromosome 2 in a process called fluorescence in situ hybridization, or FISH for short. What this involves is the addition of a DNA probe containing a short stretch of DNA bound to a reporter molecule. If it binds successfully to the target DNA, and any unbound probes are removed by washing, you can visualize the bound probes. If the reporter molecule the DNA is attached to is a fluorophore, this can be done directly due to the bound reporter molecules fluorescing. Alternatively, if the reporter is a protein, fluorescent antibodies can be added to bind to the protein and indirectly end up with the same result. Regardless, FISH can be used to visualize where on the chromosome certain stretches of DNA complementary to the probe are located, as used in this study. 2 cosmids found within 2q13, called C8.1 and C29b, were found to persistently localize to chromosome 2. Sequencing them demonstrated that C8.1 contained 2 stretches of TTAGGG repeats arranged to meet each other. In other words, in an inverted fashion. The same thing goes for what are called the flanking sequences. These flanking sequences show 95% and 90% sequence identity, with 2 different sequences found next to telomeres located at the ends of chromosomes, or pre-telomeres. This strange layout of both the telomeric sequence and the DNA on either side is, according to the authors, what you would expect to find if two chromosomes had fused head-on, put another way, at the telomeres. However, there is a second process which could have created the same arrangement, specifically a random duplication and inversion of a section of the chromosome. To rule this possibility out, the researchers next isolated stretches of DNA called subclone 817 and subclone APA813. These sequences were not part of the telomeric repeats, but flanked them. 817 and APA813 were then hybridized to yeast chromosomes which had been artificially made to include human telomere DNA. Experiments involving a nuclease called BAL3-1 and fish demonstrated that these two sequences both hybridized to 2q13, as expected. Importantly, both of them hybridized to distinct but still overlapping stretches of DNA near the tip of a chromosome. The idea that part of a chromosome duplicated itself and then flipped to create the results seen above was ruled out by the authors. This makes sense. After all, if the two blocks of telomeric DNA had come from the duplication of part of the same chromosome, you would expect the sequences to be identical. Thus, chromosome 2 consists of two chromosomes which fuse together. However, the joining together of two chromosomes at their telomeres is not the final step in merging two chromosomes, and there are still several that need to come after this. As just one example, each chromosome always contains one centromere, which I can't go into today, but plays an important role in cell division. As this fused chromosome now has two of them, one needs to be removed. Interestingly, the authors noted that previous work and unpublished data from their own study pointed to an ancient former centromere at a different band called 2q21. So, to summarise all of this, by using fish to hybridize cosmids containing sections of the human genome to chromosome 2, researchers showed that a section of chromosome 2 called 2q13 contained two sets of telomeric DNA, which is usually found at the tips of chromosomes in order to protect them. Both the telomeric DNA and the regions flanking them were arranged in such a way that they met each other head-on, in the same way that two separate chromosomes in contact at their telomeres would do. Because the possibility of a smaller change involving part of the chromosome having duplicated and then flipped was ruled out, researchers concluded that chromosome 2 consisted of two once separate chromosomes, 2A and 2B, which had merged together. The existence of a former centromeric region would also support this. It is apparently difficult to say how often such fusion events have occurred in evolutionary time. This is because very old fusions cannot be as readily spotted, given that the former telomeres and pre-telomeres will mutate over time. So, from an evolutionary standpoint, we've been fortunate enough to live recently enough to still be able to detect this ancient chromosome fusion. That's where we'll leave the discussion for today. In the meantime, thank you all for listening. Feel free to get in touch at the show's email address for any questions, comments, or topic suggestions for future episodes. Until next time, have a great week, everyone.